Please subscribe our channel Education Point and press bell icon to get notification of new educational videos.
Hello everyone, welcome to lecture number 119, and today I am going to discuss the characteristic, very important characteristic of protein. So I will be discussing one by one. First is when it is subjected to hydrolysis, either in presence of enzyme or mineral acid like hydrochloric acid or sulfuric acid, we get the final product amino acid, and sometimes intermediate product that is called dipeptide or peptide. Secondly, it is colloidal in character. The characteristic of protein is colloidal because it is complex and large molecule, and that's why proteins, they have hydrophobic in character because it can absorb the water and some other substances also. And protein is amphoteric in character, means it is having both an acidic as well as basic, so it can react with acid as well as with base. And if any compound which has that property of reacting with both acid and base is called amphoteric character. So protein is having amphoteric character. And then thirdly, isoelectric point. Isoelectric point means it is a critical pH. I have already told you in amino acid chapter when I was discussing isoelectric point. So isoelectric point of protein means it is a critical pH at which the ionization of amino group as well as carboxylic group, they are at equilibrium or almost very minimum ionization is there. That is called isoelectric point, and the isoelectric point will help you to isolate and purify the proteins. So here I am discussing you one by one. First number is, that is hydrolysis of protein. On hydrolysis, protein gives amino acids or intermediate molecule, that is peptides. Hydrolysis can be brought by heating with dilute acid or enzymes. The enzyme which is used to hydrolyze is called proteases. Number 2, colloidal character of protein. Due to complex molecular structure and dimensions, proteins are colloidal in character. They are hydrophilic in nature and bind water up to 0.3 to 0.4 gram per cubic centimeter. Number 3, amphoteric character of protein. Like amino acids, proteins are amphoteric in character, capable of reacting both with acid and base. Number 4 is isoelectric point. Just like amino acids, for each protein molecules there is a certain pH known as isoelectric point at which its ionization is at equilibrium. For example, the milk protein casein has an isoelectric point 4.6. This character of protein often made protein to be isolated and to be purified. So now I am discussing denaturation of protein. When protein is heated or in presence of strong acid, it loses its all chemical and physiological property. That is called denatured protein, and phenomenon is called denaturation. For example, when we are heating the egg, coagulation will take place, and that coagulation becoming white in color and solid, that is denaturation of protein. Then other, that is optical activity of protein. Proteins are all optically active and they are levorotatory. The optical rotation is levo. And now I am going to tell you some 3 important color reactions or color test of protein. First is Butte Test, and then Ninhydrin Test, and third one is xanthoprotein test. That is, Butte Test means here copper sulfate alkaline solution is treated with the protein, and it gives you the violet or blue color. Then Ninhydrin, in Ninhydrin means Ninhydrin itself, when it is treated with protein, it will give you dark blue color. It is sensitive reaction, and that indicates the presence of protein. Xanthoprotein test means concentrated nitric acid. When it is treated with protein, especially aromatic one ring, it gives yellow color, and that indicates the presence of protein. And you might have noticed, when you are working with concentrated nitric acid in your lab, chemistry lab, you might have noticed that whenever the concentrated nitric acid is touched to your finger, you will get a yellow stain on your finger, and that yellow stain means that nitric acid has done the nitration of your skin protein, aromatic ring, and that's why you get yellow color, and because your finger itself is protein. So here, denaturation. Protein undergo changes in behavior when heated or treated with strong acids or bases or certain reagents, and therefore loses their physiological properties. The protein so modified are often referred as denatured protein, and the phenomenon is called denaturation. For example, coagulation of egg by heat. Optical activity. All proteins are optically active, and they have L configuration, means Leo rotatory. Test of protein. First is Butte test. When alkaline solution of protein is treated with copper sulfate solution, a blue or pink color indicates the presence of protein. Second, B is Ninhydrin test. It is very sensitive test involving the reaction of pyridine solution of the protein with Ninhydrin. Deep blue color indicates the presence of protein. And number C is xanthoprotein test. It involves the reaction of protein with concentrated nitric acid, and there will be formation of yellow color. That indicates the presence of protein. So with this, I will stop here, and in next lecture I am going to discuss very interesting topic and some lot of new information that will be starting from 120 lectures. That is nucleic acid, DNA, RNA, the hereditary material. It's a very interesting topic. Thank you very much.